Hello, and thank you for attending today's webinar, Cracking the Code on Complex Proteins, a panel on process innovation and scalable production, presented by AGC Biologics and Fierce Biotech. I'm Andrea, and I'll be your moderator for today. And finally, we'll host a Q&A session at the end of the webinar. Feel free to submit your questions at any time using the Submit Questions window. All right, let's dive right in. In today's presentation, we'll be exploring the growing demand for complex therapeutic protein manufacturing. The bispecific market alone is projected to grow exponentially in the next few years, and these atypical molecules offer the potential to transform clinical outcomes. However, their complexity presents specific development and manufacturing challenges that require rethinking some of the standard processes. With that, I'd like to introduce our experts on today's subject from AGC Biologics, a leading global CDMO providing development and manufacturing services for protein-based biologics and advanced therapies. So joining us today is Kayla Bean, Senior Manager, Technology and Innovation. Gilbert Martinez, Process Development Fellow, Analytical and Formulation Development, and Kevin Dornfeld, Principal MSAT Engineer at AGC Biologics. Our speakers will be presenting key strategies for developing standardized and scalable processes, from early development through cGMP production, that mitigate risk and consistently deliver high-quality, stable molecules. Our first speaker, Kayla Bean, will present strategies for developing stable, high-yield cell lines using transposase technologies. Kayla, the floor is yours.
Thank you for the introduction, and thank you for everyone who's attending. So cell line development is very important for drug discovery as well as biopharmaceutical manufacturing. And the strategy that we take in cell line development can directly impact cost, timelines, and the probability of success for that program. And within this strategy, there are several decisions that you need to take depending on the platform, the vector choices that you use, and how you integrate the gene of interest that you're hoping to express. And that's what we'll be speaking about a little bit today. In the bottom right-hand corner, we'll be discussing kind of what we did in the past. You'll see what random integration looked like. Some of the issues that were with random integration in the past had some instability issues, whether it was for concatemerization during integration or truncated expression, gene silencing over time. These issues ended up having long-term instability with manufacturing in some cases. Technology has advanced and semi-targeted integration was adopted in most of industry, which ended up increasing the expression consistency and reducing some risk for that instability. We have employed the leap-in transposase platform from Atom. This platform utilizes a cut and paste technology at the ITR regions. This transposase prefers active open chromatin for the insertion of the gene of interest in these high transcriptionally active areas, which gives us potential for high expression of our protein of interest. Supporting high expression, more integration means more stable expression here, and this increased efficiency in integration gives us more copy number integration within the cells. More copy number gives us potential for higher expression and higher productivity, more productivity per cell, which increases our overall titer. This is very important not only for titer, but also the idea of getting a full insertion of the gene cassette. And this is very important that we can ensure the correct ratios of our insertion, because for these complex biologics that we're looking at, it's very important to ensure that we're getting the right ratios so that we can get the proper formation of the molecules that we're looking at. Moving into our next slide, we can see just some of the examples of how we've seen the increase in our titers. We've had several examples in some of programs we've done, as well as in some of our tech dev programs between stable pools, as well as some of our clonal derivatives that we've seen as much as 10 times increase with this technology that's helped enhance our programs. And again, not only with our stable pools, but as well with our clones. And as we were talking earlier about that efficiency and integration, it helps with our stable pool levels by giving us less diversity. versus the random integration where we had a high level of diversity. So what this means is we have more comparability between our pools and our clones. This is the idea that we can have a little bit of early on development. Maybe we're able to assess the pools at an earlier time so that we can maybe see if there's any issues that may cascade later on in a program. And this early on ability to assess the molecule may give us increased efficiency, may give us some reduction in timeline. And also along the same level, we're also getting very increased titers, but we also, again, want to look at reduction in timelines. How do we do that? We're also implementing high throughput automation, screening for our clones, and using data-driven selections. So we're doing that using the Beacon instrument, using on-chip assays to look at titer, as well as other cell attributes. So overall, the point in the summary here is that as the molecules get more complex, CLD can't stay traditional. We have to use these data-driven and technology-driven solutions in order for CLD to become more precise, more automated, and more predictive over time. So thank you.
Yes, thank you, Kayla, for your insights on the shift from a traditional empirical cell line development process to a high throughput, strategic, and data-driven engineering process that is highly specific for each complex protein. So continuing our discussion, Kayla, can you tell us what are the top concerns in cell line development for complex molecule development, and how should a CDMO navigate those challenges?
Yes, what we're seeing for these complex biologics, bispecifics, trispecifics, we're really worried about, of course, of genetic instability. How are we going to scale these molecules? But also looking at mispairing, again, making sure we're getting the proper assembly that we're looking for and that the molecule is functional and we're getting the highest percentage of that functional configuration that we're looking for. And on the CDMO side, really looking for high throughput automation, what types of expression platforms are being used, and really looking at the analytical workflows that we can use to assess manufacturability early on so that we can really come up with the best cell line to move forward in the process. Thank you.
All right, thank you, Kayla. We really appreciate your insights on that. And that's actually a great segue to our next speaker, Gilbert Martinez, who will be discussing analytical workflows and how his team uses high throughput technologies to quickly identify risks and select high-performing clones. Gilbert, please go ahead.
Thank you for that introduction. Thanks for everybody tuning in. Yeah, Kayla brought up some very good points in her discussion. So first is that we're getting higher titers. And second is that we're shortening our timeline. So everything needs to be done faster. And this poses particular challenges for quote unquote complex molecules. Whereas for regular monoclonal antibodies, there's a long history in the industry of developing antibodies. So we have some good processes and good methods in place to do that and to do analytical monitoring very fast and efficiently. However, some of the molecules that we're seeing are quite complex. There's no one molecule that's exactly alike. So this poses different risks compared to antibodies, and it means you have to identify these different risks because it's not going to be the same for every antibody. In addition, the higher titer, what's going on when you're having high titer clones is that they're expressing the cell at very high levels. So you're taxing the cells that are creating these molecules. That could lead to problems in the correct folding, could lead to post-translational modifications. It could lead to a lot of different issues due to that very high titer and rapid turnaround of proteins being generated within the cell. So in order to choose the best clone, what we've done here at AGC is start adopting more and more data into our CLD flows. And one of the things we look at specifically is like the useful yield of the clones. So you could have a high titer, 10 grams per liter, but if 50% of that is aggregate, you're not getting the full 10 grams per liter out of that cell line. And furthermore, if you're getting 50% aggregation, that's going to pose risks to all the downstream processing that needs to take place. So there's a very good reason to start looking at analytics more carefully early on. And that's exactly what we're doing here at AGC. And in order to keep our timelines, we keep our cells growing. So we don't really have any window to stop and get good analytics in a long period of time. So we have to do the good analytics in a very short amount of time. So we've adopted a lot of automated methods, particularly automated chromatography and sample preparation. So we can purify our top 30 clones from our workflow within 24 hours. And we have short targeted methods to assess product quality in shorter and shorter time. So within 48 hours, we could have all of our data collected. And using automated processing methods, we can start processing those data sets in real time or have very rapid turnaround processing. So we can get routinely 30 clones and PQ analysis within 72 hours. That lets us keep our cell line going so we don't have to delay any decision points. And then we could cut off at our top 8 or top 5 clones, however we want to proceed with that. And one of the things we've adopted in this process and getting this increased amount of data is to develop a comprehensive scoring assessment system in place. And we take in all of the data from all of the assays that we identified as worth pursuing, as well as cell culture, cell viability, titer, and other metrics. And we combine all of that into a scoring system that we could sort. And as a team, our upstream colleagues, our downstream colleagues, our analytical colleagues, and cell line development colleagues, we all get together and identify what we think are the most critical quality attributes to look for. And we can rank and score our different clones. So we're not just relying on a single titer value, for example. And this has come into play on a couple of our recent campaigns where our highest titer clone ended up being something that we did not pursue because it had high risks associated with that early product quality data. And at least on one case, we had a high risk clone that we carried along in the process just to see if, our old thoughts of titer only, was the most important. And in fact, that clone did not survive our top 2 clone selection. So our scoring system does point out risks that we can identify earlier on, and we don't have to wait for either stability or final clone selection. So on the next slide, I'm going to talk a little bit about how we go about getting our analytics set up to support the rapid turnaround times. It starts for us at AGC by identifying the molecular risks. The Seattle site at AGC Biologics has a lot of structural biology experience. So we really understand molecules here, both on the sequence level and on the structural level. So our team goes through and looks for all of the main molecular risks, such as sequence modifications or structural risks associated with the molecule. And again, these complex molecules can look completely different than mAbs. You could have fusion proteins, molecules not even associated with antibodies. So you really need to spend this time really identifying early critical quality attributes that are going to be important in your cell line development clone selection. So once we identify the sequence and structures we want to look at, we have a lot of different methods that we could use. We have advanced chromatography. So we use a traditional 1D chromatography, as well as advanced two-dimensional chromatography. And the two-dimensional chromatography gives us the ability to look at multiple attributes at the same time. It also gives us a chance to look for peak characterization. So if we see, a high molecular weight species or a low molecular weight species, we could identify what that impurity is very early on. We also have some advanced mass spec methods that we're using. We have a new instrument that has very high resolution and advanced separation techniques. So we can identify a lot of different post-translational modifications. And one of the things that we're seeing more often is sequence variants. So we can start looking at sequence variants in our early clone selection. And in fact, in one of our recent campaigns, one of our top 8 clones was rejected because we saw high levels of sequence variants being incorporated into the molecule. So again, just to summarize, one thing we do here at AGC Seattle is rapid analytics and we target our analytics and the targeting of the analytics, what you're looking for, why you're looking for it is very crucial because that gives you the ability to make more informed decisions with all of this analytical data that we're collecting. So we're collecting a lot of analytical data, but we're doing it with a purpose and an intention of choosing the correct clone for manufacturing. So that's what I have to say. So I'm going to bring back the moderator.
Thank you so much, Gilbert. It was really interesting to take a closer look at the automation and advanced analytics that are providing such in-depth data around clone fitness. Enabling rapid decision-making seems to be critical in complex molecule manufacturing. So furthering our discussion then, maybe you can tell us what are some of the shifts you are seeing in the industry and how are analytical advancements helping set the new standards for complex molecules?
Yeah, so what we're definitely seeing, two things that we're definitely seeing is just the sheer amount of complexity in these different molecules. No molecule is the same. We do have some bispecifics that are very much similar to the regular monoclonal antibody architecture, but we have some that are much different than that. And we also have molecules that aren't antibody related at all. So the level of complexity of the molecules we're seeing is something that I've noticed coming to us from proposals. The other thing is these very short, aggressive timelines, as you discussed. Everybody wants it shorter and shorter timelines, and that makes sense. So we have to adapt and keep up with that. So like our cell line development, we really need to keep everything moving along and move right into upstream development as soon as possible. If we stop to freeze a cell line, for example, that's going to delay the timeline a week or 2 weeks to start growing and propagating those cells. So we have no time for that. So we really are trying to target our analytics and taking a minute early on to identify what we should be looking for, what's going to help us make the best, most informed decision, and what analytical methods we need. So I feel like that's where it's going.
All right, thank you so much, Gilbert. Now, I know that atypical molecules such as bispecifics and trispecifics present some unique challenges when it comes to purification. And Kevin, I believe you plan to share with us then how titers can present different challenges with complex molecule manufacturing with respect to purification and downstream processing. So please go ahead for us, Kevin.
Yes, thank you. And thank you to everyone joining today. Yeah, so the new complex molecules, bispecifics, trispecifics, Fabs, they can really challenge our standard monoclonal antibody purification platform process where we need to now tailor development to the unique structure of the molecule because of decreased stability, higher levels of aggregates or mispairings, and also lower than the typical step yields for the operations of downstream. And this is where our experience with many different types of molecules across different resins and different chromatography modalities, including affinity, ion exchange, cation exchange, mixed mode, and hydrophobic interaction chromatography really allow us to help narrow down which resins are going to be best suited for this particular molecule and include those in our screening studies and narrow our focus to the most favorable buffer operating conditions, which will allow us to optimize those processes. And this really helps us just to narrow our focus to limit the number of experiments to complete, to eliminate or reduce rework where we go down pathways that don't lead to our final process and shorten our timelines. And so next, I'd also like to summarize some work we've done with a case study where we optimize a bispecific process that it initially had unacceptable levels of product variance or product-related impurities. And I'll be focusing on our chromatography steps where we evaluated affinity resin that was not part of our platform process at the time, as well as optimizing the cation exchange step and the addition of a fourth chromatography operation for polishing using hydrophobic interaction chromatography. And so for each of these steps, I'll focus on what the process step is, the rationale for how we go about development of that unit operation, the purpose of that process step, and then ultimately why we chose a particular resin. And so for first step here with a bispecific, it's not always so much that the affinity or single bond strength can be the most effective for separating out your product of interest versus product-related impurities. We want to look at the avidity of the resins that you'll base off of your particular molecule. We're developing basically across multiple bond sites and looking at the total bond strength. For our example here, we were first looking at a traditional protein A resin that bound to both the FC and the VH3 region, but we found that it did not separate our mispaired homodimers from our target molecule. And so we evaluated a CH1XL resin that was able to give us the product quality that we need to reduce the burden further downstream for purifying this molecule. And then next we have a ion exchange step that's pretty typical to have a flow through an exchange step that utilizes single-use membranes. And here we just chose the membrane that gave us the highest HCP, host cell protein clearance. And that was a cytosine Q for this step. And then next, we need to further improve our product quality profile as far as aggregate levels and charge distribution. And so we're looking at several kind of exchange resins here that we have most familiarity with. And then we chose a resin based off initial screen studies, the porous XS. And then we need to further fine tune that operation by optimizing the buffer conditions and the collection criteria around the elution for this molecule. And by doing that, we're able to achieve both our product quality targets and maintain a high step yield within our target. And then finally, we found that although we were able to achieve pretty high quality after a third step, we wanted to further polish this process with a fourth chromatography step. And sometimes here you may look at a hydrophobic interaction chromatography HIC step or multimodal chromatography step that has both ion exchange and hydrophobic properties. Here we end up going with a hydrophobic interaction chromatography step, capital butyl impres. And then we further optimize this process through the low conditions, the wash conditions, and optimate the elution conditions for this unit operation. And with that, I'll send it back to moderator for questions. Thank you.
Yes, thank you so much for that dive into why standard MAB purification methods might not work for more complex molecules. It was interesting to understand a little more how you optimize technologies to help maximize purity and yield with these projects. I did have one follow-up question. So are there any new purification technologies in development to support the growing demand for complex molecules?
Yeah, we have a few tools at our disposal. First, I think the library of resins, especially around affinity resins, is growing. And that really helps for bispecifics where, say, a traditional protein A, a MAB select, or other resin that just binds to the FC region of a molecule will not give you the product quality that you need. So looking at VH, we have different resins that will bind to the VH3 resin, VH3, or the antibody CH1, or a protein L with a kappa light chain or lambda light chain. Or also that we have molecules that are sensitive to low pH. So there's some resins that we can use that will allow us to elute at a more neutral pH. And you can also kind of going back to our case study where originally we use a CH1XL resin. There are now competitor molecules out in the market now that have increased stability and higher dynamic binding capacity compared to previous generations of resins, such as the one we chose. So we have more options now as well for having a standard cleaning regimen with sodium hydroxide. There's also greater utilization now of using in silico process modeling software to help us reduce the number of experiments required for process development, because we wanted to really work with shrinking timelines. And with a complex molecule, you could see yourself going into having a high number of experiments to optimize the process. We wanted to limit that time and that number of experiments. So with having process modeling, we can characterize a column and a resin with the system, and then for your particular molecule, run a limited number of calibration experiments that will help us then develop a model that we can optimize our process. So that'll really give us some gains in separating challenging product-related impurities and all while doing that, achieving our yield targets.
All right. Well, thank you so much. So we have heard from each of our speakers. Now it's time for the Q&A portion. If you haven't submitted your questions yet, please do so now. And I do see a number of questions already. We will try to get to as many as possible. So for our first question, how do different molecular architectures impact expression, stability, and scalability? Looks like maybe that one could be for Kayla.
Um, yeah. So lots of ways. How the molecule is branched, symmetric, asymmetric. I think Gilbert spoke to this a little bit. Gilbert, you may have a lot to add here. The mechanism or mechanism of action, how this molecule is working, the FC effect here on the function. It'll affect a lot of things. How the format of this, it may affect downstream. It'll affect the scalability strategy later on. Gilbert, do you have things to add here? I know you kind of touched on this a little bit.
Yeah, so it depends on the level of the complexity.
Yeah.
If we do have some that have non-covalent interactions with them, so that is something that you need to look at and look for as well, because you could be expressing them, but if they don't come together, it's not going to be the fully functional molecule that you need.
Yeah.
And some of these molecules are very complex, so they have linkers in between different domains. And those linkers are not very stable. They're prone to proteolysis. So we also have to look at potential truncations as well.
Yeah.
All right, thanks for those responses. And another question here. How do your analytical methods directly support the downstream purification team in particular? Are you developing rapid in-process control assays that allow them to make real-time decisions during a purification run for a complex molecule?
Yeah, so we definitely support the downstream process development team throughout the project. And this again starts with knowing your molecule. When they're complex, you really need to know what's what the risks are. And each process step, there's certain conditions. What does the molecule look like in those conditions? So if you're going to low acid for viral inactivation, which is something we see a lot of molecules are not very stable there. So we need to find ways to assess that, whether it's through size exclusion chromatography, or what we're doing now is including more and more mass spec in our development, because with the high throughput methods we've gotten in place for CLD, cell line development activities, we can translate those to others. So we are adding more data to the downstream process, but it really goes to understanding your molecule, understanding what each step, what risks can happen to the molecule at each individual process step along the way. But we definitely support the downstream process throughout its entirety.
All right, thank you for that. Another question, could you talk about some of the current or emerging technologies around process intensification? I think maybe Kevin, you could take that one for us.
Yeah, I can take that question. So in recent years, there's been several single-use affinity membrane products coming on the market, and there's more on the way. And with these, it allows us to utilize the full resin lifetime of a protein A ligand in a single batch. Because typically with a clinical manufacturing, you may only be running this operation a few times, maybe 10 cycles, and resins typically have 100, 150, 200 cycles they can run in a lifetime. So you're not getting the full use of your resin, and affinity resins typically are the most expensive part of the downstream process. And so this really allows us to decrease the cost for the capture step for a monoclonal antibody.
All right, another question here. At what point in discovery or early development should CLD strategies start influencing decision making?
I think I'll take that one. As early as possible, start assessing those risk factors as soon as you can. Each of these genes of interest or what you're wanting to express, these sequences have very intricate details, as we were just talking about the architecture. You know, the amino acid sequences have risks factors involved with those, looking at multiple different variants. Then, of course, how are we going to express those? What are the vectors we want to choose to put those in? What are the designs of those vectors? Looking at signal peptides, chain ratios, what promoters do we want to use for each of these? And, you know, long term, what's the requirements for the therapeutic application here? And thinking very early on about what the end goal is when we get started, so that we don't have to, you know, redo something in the middle. Just getting all the information up front, thinking everything through clearly, and making all of those critical decisions as soon as we can.
Yeah,
All
can
right.
I add
So,
something
yeah, go ahead.
to this? Yeah, I think Kayla is very is spot on. We want to do this as soon as possible, and any recommendation to some companies about to go into development, anything you could do to support manufacturability is helpful. Like a low pH hold, or you have high ionic strength in some condition, or no ionic strength in some conditions. How is the molecule? What is molecule stability in that regime? Like, is it going to pose a risk for downstream? Because if we're just using our platform and we're missing a key set of data, we could lose time on that. But if you could say, it's not stable at low pH, or you need this minimum amount of salt, that will inform our downstream process, and that's just very valuable information, especially if you want a really tight timeline. So the more data you can get early on, the better.
Right. Or even so, telling us that you have no idea, so that we can even look at that early on, right? So we can start assessing where are those critical points early on. So, yeah.
All right. And Gilbert, here's a question that I think might fill out a little bit more about what you were saying just now. But can you share an example of where an early formulation decision successfully prevented a stability issue that could have derailed a project in later stages?
Yeah, so pH stability is definitely an issue. In the past, I've worked on a molecule that had it was reversible aggregation at low pH. So it was aggregating, but you could correct it later on. But it posed some significant challenges with the purification strategy, because we were seeing almost all aggregation. So you might lose that in the downstream process. So by identifying, like, okay, if you want to use this low pH, you absolutely have to increase the ionic strength to a certain level in order to break up those aggregation interactions. So that helped in the past, and the low pH is definitely something we're concerned about for all of our viral inactivation. The other thing that I would like to throw out there, it's about the state of the drug substance. So even after we purified the whole process, if you don't do some analytics to get the glass transition temperature, so that's a measure of when the molecule starts freezing and getting into an ordered frozen state. That is very important. You want to be as far away from that as possible. And we have one client come to us that was storing their molecule right about their glass transition temperature. So when you opened a freezer, for example, it might be going down below that temperature, even for a short period of time. But if you have multiple cycles like that, it can cause problems. And in fact, in one of our programs, we did see that, and we were able to track it down to the freezer being open, because it was just stored at a very inconvenient temperature. So looking at that, and then the glass transition is going to be dependent on the molecule and the formulation. So that is something to keep in mind.
Okay, another question. What are the most common purification pitfalls that you encounter with complex proteins, and how do you proactively design processes to avoid them?
Yeah, I think tagging along on the discussion for product stability is ensuring that you have the right package of product quality data for your process intermediates, where especially here where our stability between operations isn't guaranteed. Uh, at manufacturing scale, the processing takes longer than at a bench scale, where you're say when you're doing viral activation, it's not instantaneous. That way, if you say your hold time's going to be 60, 90 minutes, that process actually takes longer. That can be 3 to 4 hours because there's time for how you're titrating down to your low pH, the time that it takes to uh, pump your product from the viral activation tank or bag into its hold container, and then beginning neutralization. That all that time is not necessarily taken into account for during your official what you're claiming as your viral inactivation hold time. So you need to have that data, understand your product stability for the pH range you'll be operating at. And then in addition to that, just when you start your development process, maybe not taking into account the specific structural properties and stability of your complex protein. If you try to go down the route initially of uh, this development process, just like a standard map, you may have some rework to optimize your process, and that just it'll increase the time that you go from bench scale to running that first GMP run.
All right, we have a lot of questions coming in and time for a few more. So the next question, how do high throughput screening technologies change the way that sponsors evaluate candidates?
I think I can take that. Gilbert, you may be able to take a little bit of that too. It allows us to look at several different things at once, from productivity to quality. Several of these attributes really help us figure out the best candidates moving forward so that we don't have any late stage surprises, as Gilbert was mentioning earlier, and we can keep those compressed timelines and higher probability of success. Gilbert, you want to add?
Yeah, sure. I think having more data lets us make a more informed decision, especially if you tackle your analytics with intent. Don't just do analytics because a certain method might not give you useful information. It might not identify any risks. So you really want to tailor it to that. But the high throughput, yes, we absolutely need that to keep the timelines in place. There's no way we could do this manually without overworking our team members and causing a lot of stress, and that's not good for anybody. Makes it very hard to meet those timelines. So the automation, really helps our staff, but it just speeds things up so much faster. And the other thing that it lets us do is handle multiple projects, right? If we're trying to do all of this manually, we'd have to stagger them even like significantly. But with automation, we could do things back to back, and that's not causing with minimal sample preparation effort needed. So it gives us a lot more data to make more informed decisions, but lets us handle more programs at the same time without risking the success of the timelines.
All right, well, we have time for one more question. So for this last question here, what CLD capability do you believe will be the most critical over the next 5 years?
Yeah, automation, using potentially AI to help us get through more of this data that we're starting to generate, looking at more and more of these larger data sets to translate the best predictions, how to take the best actions quicker, looking forward, real-time analytics, making sure that we have predictive modeling quicker so that we can identify manufacturability and scalability, and just keeping up with industry and automation. Yeah.
All right. Well, thank you so much for your contributions to this discussion today, and thank the audience for asking such thoughtful questions. That is all the time we have for today. If we were not able to get to your question today, we will do our best to follow up after the webinar. As a reminder, a recording of this session will be available on demand within 24 hours. Thank you for joining us, and we look forward to seeing you next time.